Nous on fait de la science. Nous on travaille pour la science. Pour la science.
Welcome everybody. Chapter 6, the mitochondria. And this chapter is a bit special because unfortunately Walter Pfaller died last year. And sorry for that. My mic fell. He died last year, and so this is the first chapter really without his input, but some of it's actually about his research as well. So I hope I've achieved my goal here. So it's chapter 6, the mitochondria. As biological science progressed from macroscopic to microscopic investigations, it became clear that the true Frankenstein's monster was the very unit of complex life, the eukaryotic cell. The chimeric entity formed from the fusion of ancient disparate lineages, housing a semi-autonomous organelle, the mitochondria, which acts as the primary governor of life, aging, death, and identity. The recognition that the cell might be a composite of independent organisms did not begin with the advent of molecular genetics, but instead with the meticulous histological observations of the late 19th century. In 1890, the German pathologist Richard Altmann published the Elementa Organismen, a transformative tome that described ubiquitous bacterial-like granules found in nearly every cell type. Altmann proposed that these structures, which he termed bioblasts, were the elementary units of life, possessing both metabolic and genetic autonomy. He envisaged a cellular landscape where these bioblasts functioned as autonomous living beings residing within a larger host. Altmann's work was met with intense skepticism and professional hostility. His enthusiastic spirit was eventually dampened by scathing criticism from his peers, including Professor F.P. Moll of Johns Hopkins, which led Altmann to become a recluse, earning the morbid nickname the Ghost before his death in 1900. The scientific community of the era sought to distance itself from the radical implications of elementary organisms living within the cell. Consequently, in 1998, Carl Benda rebranded these granules as mitochondria, a term derived from the Greek for thread granules, effectively reducing them to mere structural components of the cytoplasm. The linguistic shift successfully neutralized the threat of mitochondria autonomy for nearly 80 years, framing the organelle as a subservient utility rather than a co-equal partner in life. The suppressive nature of early 20th century biology towards mitochondrial independence mirrored the demonization of Frankenstein's creature. Just as society was reluctant to accept a man-made sentient being, the scientific establishment was reluctant to accept that the human species was a product of a primordial microbial merger. It was not until the work of the American biologist E. van Valen in the 1920s that the concept of symbiotism was expanded, suggesting that mitochondria were originally free-living bacteria that had entered into an obligatory partnership with a host cell. Valen's claims, including the controversial assertion that he could culture mitochondria outside of cells, were largely ignored because the tools of the time were insufficient to prove the existence of an independent mitochondrial gene. The modern renaissance of the embiotic theory is inextricably linked to the persistence of Lynn Margulis. In 1967, writing under the name Lynn Sagan, she published on the origin of mitosing cells, a paper that had been rejected by more than 15 journals. Margulis argued that the eukaryotic cell was a result of a unified theory of eukaryogenesis, where at least three fundamental organelles, mitochondria, photosynthetic plasmids, and flagella, originated as free-living prokaryotes. While a proposal regarding flagella, also called undulhipodia, remains controversial due to a lack of associate genome, her advocacy for the bacterial origin of mitochondria and plastids fundamentally changed the landscape of evolutionary biology. Margulis posited that about 2 billion years ago, a primary anaerobic host cell, likely an archaeon, ingested a aerobic protobacterium. Instead of digestion, a symbiotic relationship formed. The host provided protection and nutrients, while the endosymbiont provided a mechanism to neutralize toxic oxygen and convert it to vast amounts of energy. The event was not a minor adaptation, but a radical leap in complexity occurring perhaps only once in 4 billion years. The rare event hypothesis suggests that the fusion of a methanogen and a proteobacterium was so statistically improbable that it represents a unique bottleneck in the history of life, potentially consigning much of the rest of the universe to nothing more than bacterial slime. The discovery of the mitochondrial DNA in the 1960s provided the literal spark of evidence required to validate Margulis's claims. The genome, though reduced over eons, remained distinct from nuclear DNA, appearing as a circular molecule that replicates independently and lacks the histone proteins characteristic of eukaryotic chromosomes. This genetic otherness within our cells is permanent monument to the chimeric nature of the human organism. As cellular respiration was localized to the mitochondrion, the powerhouse of the cell metaphor became the dominant narrative in biological education. Popularized by Philip Siekewitz in 1957, the analog compares the organelle to an electricity generating station that produces useful energy, also called ATP, for the cellular city. This bioenergetic function is governed by chemiosmotic theory proposed by Peter Mitchell in 1961. Mitchell's paradigm-shifting work demonstrated that mitochondria generate energy not through traditional chemical reactions alone, but by creating an electrochemical gradient, a literal electrical potential across the inner mitochondrial membrane. The magnitude of this electrical spark is staggering when viewed at the microscopic scale. Gram for gram, mitochondria generate more ATP than the sun, utilizing a proton motive force that drives the ATP synthase turbine. The process, termed oxidative phosphorylation, dictates our every breath. We inhale oxygen specifically to satisfy the requirements of our mitochondria, and the carbon dioxide we exhale is the waste product of their tireless metabolic labor. The physics of the process can be modeled through the relationship of the electrochemical potential. The spark is the biological realization of the Frankensteinian dream, a controlled flow of energy that brings inanimate matter to life. However, the reduction of mitochondria to a mere power plant obscures its role as a dynamic signaling hub and a central processor of cellular information. Okay, and this bit now brings in Walter Pfaller. While the powerhouse metaphor suggests a static utility, the research of Walter Pfaller at the Medical University of Innsbruck reveals that mitochondrial governance is a highly dynamic and structural affair. The scientific cornerstone of this quantitative approach was Pfaller's 1982 habilitation thesis titled Structure-Function Correlation on Rat Kidney, Quantitative Correlation of Structure and Function in the Normal and Injured Rat Kidney. This work moved beyond qualitative description to establish a direct mathematical correlation between the volume of mitochondria and the surface area of the inner cristae membranes, and the specific metabolic work performed by different segments of the nephron. Most notably, the ATP-driven sodium reabsorption in the proximal tubule, i.e. sodium-potassium ATPase. This research provided definitive evidence that biological function must be physically consistent with, if not derived from, the precise scaling of the subcellular architecture. Pfaller utilized meticulous electron microscopic morphometry to demonstrate that the internal architecture of the mitochondrion, that is the cristae, is highly adaptive. In studies on rat kidney cortical nephrons, he discovered that a high potassium diet induced a staggering 200% increase in the density of cristae mitochondrials. The structural expansion represents a massive scaling of cells' energetic governance to handle the metabolic work of potassium adaptation. Conversely, he showed that external insults, such as halotene anesthesia, could cause immediate mitochondrial swelling and a 30% loss of inner membrane surface area, effectively downsizing the organelle's functional capacity. Furthermore, Pfaller's research in renal epithelial cells, including LSCPK1 cells and OK cells, identified a significant metabolic switch that occurs when cells are moved from the body into an artificial culture. As cells mature into a monolayer in vitro, they often dedifferentiate, losing their complex apical microvilli and suffering a substantial reduction in mitochondrial volume. The structural retreat is accompanied by a shift from oxidative metabolism to high rates of glycolysis, a phenomenon where the Frankensteinian assembly of the cell reverts to a more primitive anaerobic metabolic state. This research highlights the corner dilemma as we attempt to maintain life outside the body. The internal governors of that life, the mitochondria, may seek and metabolically abdicate, threatening the identity of the very tissues we seek to preserve. Mitochondria function as cellular processes that receive the integrate information from the environment, producing signals that influence the entire organism. They sit at the confluence of metabolic pathways, adjusting the nutrient availability and preventing the buildup of toxic products. One of the most critical aspects of this governance is the reduction of cell fate transitions, deciding when a cell should divide, differentiate, or die. Recent studies highlight the executive role of the mitochondria in stem cell differentiation. For example, high activity of mitochondrial enzyme OGDH, which is a key factor in the TCA cycle, supports energy production and growth in absorptive cells, while low OGDH levels lead to an accumulation of alpha ketoglutarate, which acts as an epigenetic signaling molecule, pushing cells toward secretory identity. In the mitochondrion, this governor, wait one second. Sorry, I forgot where I was. I think it was here. If the mitochondria is the governor of the eukaryotic life, it is also the judge and executioner of death. One of the most significant surprises in the 20th century cell biology, was the discovery that mitochondria are central points of the convergence for signals inducing apoptosis, otherwise known as programmed cell death. This process is not a chaotic failure, but a predictable, biologically programmed phenomenon essential for sculpting embryonic shape and maintaining tissue health. Mitochondria adjust the decision to commit to death through a complex of interplay of the BCL2 family of proteins at the mitochondrial outer membrane. This judicial board consists of pro-apoptotic initiators, effectors, and anti-apoptotic protectors. When stress signals such as DNA damage or viral infection tip the balance, the effector proteins Bax and BAC form pores in the mitochondrial membrane, a process known as mitochondrial outer membrane permeabilization. Once the membrane is breached, the executive phase begins with the release of cytochrome C into the cytosol. In the cytosol, cytochrome C facilitates the assembly of the apoptosome, a 700 kilodalton multimetric complex that activates the caspase family of proteases, the biological executioner within which meticulously dismantles the DNA. The dual role of mitochondria as both the source of life-sustaining energy and the reservoir of death-dealing toxins creates a profound bioenergetic and existential tension. By coupling energy generation with cell death, the cell ensures that the two conflicting processes are generally exclusive, favoring a swift and non-inflammatory removal of damaged components. The unique inheritance of mitochondria has also enabled a different kind of reanimation, the reconstruction of human history. By tracing variations in mitochondrial DNA back through time, genetics identified a matrilineal, most recent common ancestor known as mitochondrial Eve. Living roughly 150 to 200,000 years ago in Africa, she is the woman from all currently living humans descended in an unbroken maternal line. The name Eve has led to the significant cultural and theological confusion, because unlike the biblical Eve, mitochondrial Eve was not the only woman alive at her time, nor was she the first human. She simply happens to be the ancestor whose mitochondrial lineage survived, while all other female contemporaries eventually produced an all-male generation, cutting their mitochondrial lines. Similarly, Y-chromosomal Adam, the patrilineal common ancestor, lived tens of thousands of years ago, meaning the two never met. Despite this, the concept of a shared biological mother provides a sense of species-wide immortality. In every cell of every human, a piece of this ancient woman, the mitochondrial genome, continues to replicate and function. The matrilineal continually acts as a marker of our collective dependence on the rest of life and our complicated evolutionary history. The question of why complex life took so long to evolve and why it may be rare in the universe finds its answer in the mitochondrial energetics. For the first 2 billion years of Earth's history, life consisted only of simple prokaryotes, that's bacteria and archaea. These organisms were trapped in an evolutionary rut, unable to increase in size or complexity because their energy production was limited to their outer membranes. As a cell grows larger, its surface area increases more slowly than its volume, creating a bioenergetic ceiling. The EMBO's symbiotic acquisition of the mitochondrion shattered this ceiling. By internalizing the energy production membranes with hundreds of small organelles, the eukaryotic cell achieved an energy per gene ratio thousands of times higher than its bacterial ancestors. This massive surplus of power allowed the cell to maintain a massive nuclear genome, invest in complex regulatory networks, and eventually form multicellular colonies. Nick Lane argues that because the merger between two independent prokaryotes is so statistically improbable, requiring the stable coexistence of a host and a potential parasite or prey, the eukaryote may be the ultimate bottleneck in the evolution of intelligent life. This suggests that while bacterial life may be common across the cosmos, the enormous leap to complexity is probably a fluke of history, unique to our planet. The pursuit of immortality is a common thread in human culture, and it is here that the mitochondrial acts as the ultimate clock of life. Since mitochondrial DNA genes mutate much faster than nuclear DNA due to their proximity to the high energy, high ROS environment of the electron transport chain, mitochondrial damage is a primary driver of aging and senescence. As we age, the collective executive decisions of our mitochondrial CEO begin to favor closure or downstream over growth, leading to gradual decline of tissue function. Modern scientific efforts are focused on stem cell rejuvenation and the potential for mitochondrial restoration in aging cells. In a manner reminiscent of Dr. Frankenstein's search for the secret of life, researchers are exploring whether replacing damaged mitochondria with young, healthy ones can reverse the clock and achieve radical life extension.
Ces deux murs qui parlent de microscope. Ces deux murs qui parlent de microscope. Ces deux murs qui parlent de microscope. Ça parle de bactéries parce que c'est des grands amis. En tout cas pour résumer.